Hello everyone and welcome back to Biology with Mrs. Evans. We're moving in basically to part 2 of Unit 9. So we've looked at how traits are passed down from one generation to the next. In this video we're going to look at some tools that scientists use basically to help them in basically discovering how traits are passed down from one generation to the next. So the first tool that we're going to talk about is a karyotype. A karyotype basically is a photograph of an organism's chromosomes. They are going to be different, obviously, for different individuals. For example, for a female, if you notice here on the slide, again, remember the first 22, and you can see that they're numbered. These first 22 chromosomes are what we refer to as autosomes. And then that 23rd pair, remember we said, is going to be our sex chromosomes, and that tells us whether or not an individual is a male or a female. So by looking at this karyotype for this individual, I know that the person is a male because I see an X chromosome and I see a Y chromosome. So what do karyotypes tell scientists or doctors? Karyotypes tell us two things. It allows us to identify the sex of the individual, and it also allows us to determine if there is a genetic abnormality. So again, you're going to have two chromosomes for each number, because you got one from mom and one from dad, and we know that they are, or we number them, and we know that there are certain genes on certain chromosomes, etc. So this individual is a normal male. I know they are normal because they have two for each of the autosomes, and they have an X and a Y. So that is a normal male. Individuals that have an abnormality will show where they're missing one or they have an extra. So here is a normal male that we just saw, and then here is a normal female. Notice there is no Y chromosome. There are two X's, so this individual will be a normal female. Notice they have two for each autosome. So in meiosis, in class, you heard me talk about that we were assuming that everything went like it should, that the sister chromatids and the homologous chromosomes are going to separate like they should in meiosis 1 and then meiosis 2. But occasionally there is non-disjunction, and non-disjunction means basically that chromosomes fail to separate during meiosis, and that results in an individual having an extra chromosome, which we refer to as trisomy, and then an individual could be missing a chromosome, and we refer to that as monosomy, meaning they only have one chromosome for that specific chromosome. So here are some examples of trisomy and monosomy. You don't need to know to memorize these. I'm not going to ask you what is Patel Syndrome and you have to tell me, oh, that's when an individual has an extra 13 chromosome. What you do need to know or be able to do is look at a karyotype and then tell if the individual is normal, whether or not they are male or female. So some of the pictures you're going to see in a moment are disturbing. So if you want to fast forward through those, that's fine. So Patel Syndrome. These are the characteristics of an individual that has Patel Syndrome, and you can see the baby that was born here with Patel Syndrome. So Patel Syndrome means that they have an extra 13 chromosome. So if you see here, they have 3 chromosome 13s, and everything else would be 2 for each. Alright, so this means that they failed to separate either in mom or dad, and then it created a baby that had an extra chromosome. Trisomy 18, which is referred to as Edwards syndrome. So that means that there is a 3rd chromosome here in the 18th position. So again, the characteristics of individuals that have trisomy 18, I'm not going to test you on that. You just have to be able to look at a karyotype and tell me normal or abnormal, whether or not they're male or female. Down syndrome, this is the one that you're probably the most familiar with. This is an individual that has a extra 21st chromosome. And you are familiar with some of the characteristics that we see with individuals that have Down syndrome. Typically is associated with mental retardation. Some of them can be born with webbed fingers and toes, and a lot of times they will have heart defects. All of these are caused because they have that extra chromosome. So, abnormal numbers of sex chromosomes. So, Turner syndrome means that you are missing your second sex chromosome. So, the individual will be what we refer to as XO. You can have extra X's or you can have extra Y's. So, an individual that has Turner syndrome, you can see this individual is missing their second set. So, typically they are going to be shorter in stature, and they are going to be female, because remember we talked about the SRY gene being located on the Y chromosome. If that fails to receive the signal to turn on, then the individual, even though they are carrying the Y chromosome, they're still going to develop as a female. So the individual that is that has Turner syndrome is going to be female because they're missing this second chromosome. So here's another karyotype, and you'll notice that they have an extra Y chromosome. And again, they could have 3 Ys, 4 Ys. So what they found is that some scientists did some studies with the extra Y chromosome, because we know that testosterone is a male hormone, and they found that individuals that have this extra Y chromosome, that is high for individuals that are in prison. So scientists basically did blood draws and then looked at karyotypes for individuals that were in prison, and they found that individuals, a high number of inmates carry this gene. And that makes sense, because by having this extra chromosome, that makes them have basically higher levels of testosterone, which can make them become more irritable, can become more violent, etc. So here is an individual. Again, this is going to be abnormal. This is called Klinefelter syndrome. The individual has 4 X chromosomes and 1 Y chromosome. So everything else is they have 2 of, but they have 4 X's. So they have 3 extra X chromosomes. So pedigrees. Pedigrees is also something that scientists use to help learn modes of inheritance. Pedigree is basically a graphic representation of how traits are passed down from one generation to the next. The format for pedigrees have changed over the years. Typically males are basically drawn as the squares, and females are drawn as the circles. And it used to be, if it was shaded in, then that meant that the individual showed the recessive trait, and if it was not shaded in, the individual showed the dominant trait. But over the years, some people have basically swapped that. But it doesn't matter, because no matter what, they're always going to give you a key, as you see here. And then the new version that you see that is half shaded like this, that would indicate that the individual would be heterozygous for that trait. They know that they are heterozygous. So pedigrees, again, picture representation of how traits are passed down from one generation to the next. So I'm going to share a little bit of personal information with you. So when I was 13, my grandmother passed away at the age of 60. There are 2 disorders that run prominently through my mom's side of the family. One of them is Parkinson's disease, and another one is a less common disease that most people don't know about. It's called Charcot-Marie-Tooth, after the 3 doctors basically that discovered it. They're both diseases or disorders that affect the neuromuscular part of your body. So my grandmother died at 60, so people in Johns Hopkins were very intrigued by my family because of the high prevalence of both CMT and Parkinson's. So they asked to do a study back in the 90s to help them determine whether or not Parkinson's was a genetic disorder, because previously a lot of scientists believed that it was caused by environmental conditions. People are eating foods that were contaminated by pesticides that basically caused these illnesses that they were seeing. But some doctors at Johns Hopkins said, I don't think that's what it is. I think it's a genetic disorder, and we would really like to come and study your family since there is a high number of individuals that have that. Both my great-grandparents had it. My grandmother did not have Parkinson's, but she did have CMT. So they wanted to do studies both on Parkinson's and on CMT. So what they did is they asked to come to a family reunion, and they charted whether or not individuals had Parkinson's and whether or not they did not. So they used pedigrees to help them learn how the trait was passed down from one generation to the next. And lo and behold, after the study that they did, both with my family, another family, I believe in Europe, and another family in Central America, they concluded that Parkinson's was a genetic disorder because they used a pedigree to show how the trait was being passed from one generation to the next. Alright, so pedigrees, picture representation or a graphic representation of how traits are inherited. We are going to practice using these in class, so just be familiar with what a pedigree is, and we'll practice filling those out in class. The next thing is the genome. So in Unit 7, you read about the Human Genome Project. So a genome basically represents the entire set of genes in a given species. So humans have a human genome, dogs have a dog genome, cats, etc. And a genome maps out all of the genes and where they are located on the different chromosomes. So in the lab station review that you guys did for Unit 7, you read about the Human Genome Project. Human Genome Project was a collaborative effort, as you read. It basically used scientists from, I believe, 16 different countries around the world, where they worked to map out the different genes on the different chromosomes. It was completed in 2003, and we know that humans, like I said, have about 25,000 genes on our chromosomes, and people fought this for a lot of years because they were worried that this information, if it got out, could be harmful to individuals. For example, I'm going to use me personally. So I talked about Parkinson's being prevalent in my family. So with the Human Genome Project, scientists now know exactly where that Parkinson's gene is, and you can actually now go and get a blood test to determine whether or not you have Parkinson's, because they can basically look to see if you carry the gene that causes Parkinson's. So people were worried that if your employer got this information, then they could refuse to hire you because they know that you're probably going to get Parkinson's and not be able to work after a couple of years, or that you're going to cost them a lot of money in insurance, and then also insurance companies could fail. One second. Sorry about that. So insurance companies could get this information and could refuse to cover you. And the good side to the Human Genome Project is that you could get information and be well informed about a genetic condition. For example, Parkinson's disease. As I said, Parkinson's and CMT are two diseases that run in my family, and a decision that I made when I got married was that I didn't want to have children because I didn't want to take the chance of passing that gene on to my child, because my grandmother ultimately was in a nursing home at the age of 40. She left her 3 kids at home because she couldn't take care of them. and she was in a nursing home at the age of 40, and then she died 1 week after her 60th birthday because of the disease basically destroying her diaphragm muscle. So I made a conscious decision not to pass that down. At the time, I didn't know definitively whether or not I have Parkinson's or CMT because they hadn't finished the testing or mapping of genes. So now I can go and get a blood test to determine if I do have Parkinson's, and they're still working on a blood test for CMT. I haven't gotten or have not gone to get that test result because part of me doesn't want to know, but I do have that option to know. So, Human Genome Project, collaborative effort by scientists to map out the genes that are present in humans. There's still work being done on that because more and more genetic disorders are being discovered, and they're trying to figure out, where those genes are located so that they can help individuals. And the idea is that in the coming years, you're already seeing it now in some of the literature that you're reading in current events and stuff like that, is that they're hoping that they'll be able to cut out those genes that cause these diseases and disorders and insert a normal version so that individuals can have offspring that would not have these disorders. So Human Genome, know that it is basically a map of all the genes located on different chromosomes. Multiple alleles. So far, we talked about genes that were controlled by two different alleles, dominant or recessive. So there are traits that are controlled by more than two. For example, human blood type is an example of something that is called traded or coded for by more than two alleles. Both A and B are dominant, so they would also basically be a codominant situation. O is the recessive allele for blood type, and I typically, when I do blood type crosses, I just use the A's and B's and O's in my shorthand as my shorthand because I know that O is recessive and A and B are both dominant. Another shorthand that you will see, which I don't use, but I need to teach you in case you want to use it, is just like we did with sex-linked traits. They will put it on an I for basically antigens and stuff like that. So I would be IA or IB, and then both of these again are dominant, and the recessive version is simply an I, little I. So blood type, multiple allele, and basically how you determine blood type is basically how your blood cells carry or do not carry an antigen. So if you are blood type A, that means that there is an antigen found on the surface of your blood cell. They carry the A antigen. If you are blood type B, you carry the B antigen. If you are blood type AB, that means you have both A and B antigens on the surface of your red blood cells. And if you are type O, that means that you have neither antigen. What that means is that your body, if you are type A, is going to produce type B antibodies. If you are type B, your body is going to produce type A antibodies. So that if you are ever given the wrong blood type, your body is going to produce those antibodies to destroy those blood cells. That is why it is important that you get your blood tested so that you know what blood type to receive, because if you are given the wrong blood type, it could ultimately kill you because the antibodies are going to cause those blood cells to clot. So this is what it looks like in a nutshell. So if you are type A blood, you're going to have the A antigen that you see here. If you're type B, you have this type B antigen, and if you are both type A and B, you have both. And if you're type O, notice you have no antigens. So again, if you are A, you're going to produce type B antibodies. Notice if you are type AB, you don't produce any antibodies, because if you did, you would be destroying your own blood cells. Type O blood is going to produce both A and B antibodies. All right. This is why type AB blood is what we refer to as a universal recipient. They can get blood basically from anybody. Type O blood, because they don't have any antigens, are what we call the universal donor. They're going to be able to give blood to anybody because they don't have those antigens to cause basically a reaction. And then you also know that when we talk about blood type, we talk about whether or not you are AB positive or negative, or O positive, O negative. There is another antigen that is basically the Rh factor that is a part of your blood type. So if you have it, you are Rh positive, and if you do not have it, you are basically Rh negative, so you would be O negative, etc. That means either you have the antigen or you don't. So antigens, again, are markers on your cell, and then your bodies are going to produce chemicals called antibodies that basically react to antigens. Remember, we talked about carbohydrates being ID tags. So those ID tags tell the body that, hey, this is basically a blood cell that belongs to you, and your body is going to make antibodies in case the wrong blood type ever enters your body. So that's blood type in a nutshell. We're going to look at determining blood type for offspring in class, but in a moment let me bring up a slide and we'll do a brief one here for a second. Alright, so before paternity tests ever came about, individuals would look at blood types to determine whether or not an individual could be the father of a child. So let's say a dad who is type O, alright, is going to give, is going to marry a woman that is AB, type AB. We're not going to worry about the O negative and stuff. So what are the chances that their child will have type O blood? So we're going to do the normal distributive property. So A, O, so the chances of them having a child that has type O blood would be 0. Notice that this child would have type A blood, this child would have type B, same for this child having type A, and this child having type B blood. So if the mom came to the dad and said, we have a child that has type O blood, then that indicates to, or should indicate to the mom that that child is not their child, because she cannot have a child that is type O, because she has type AB blood, and both of those are dominant. So she would either have a baby that is A or a baby that is B, or if the dad was type B, then she could have a baby that has type B blood. So let's say she gets divorced and she gets married to a gentleman that has type A, B blood as well. They want to know what are the chances of their child having type B blood. So, same as usual, distribute them through. So the chances of them having a child that has type B blood would be 25%. That is blood type. We're going to practice it in class as well. And like I said, this was how they would do paternity test before DNA fingerprinting became big for determining paternity more accurately. So be able to do these. We're going to practice them in class. That's it. Have a great day. Bye.